So good evening. So why do children and adults learn differently? That's the big question, the question of why. So can we actually paint our brain, as was seen in the animation there, with crayons? Can we actually design our brain, our own learning processes? Tonight we will get answers from renowned researchers who for decades have worked on this subject. And I'm very pleased to welcome all of you to this hall in Hamburg and all of those joining us in the live stream. I'm Andrea Grießmann. I'm really happy to welcome you on behalf of the Hector Fellow Academy and to accompany you through this evening. Now, if you're in the live stream, you can listen in German or in English. So you have the choice of two languages. Later then, during our panel discussion, everyone here in this hall or in the live stream can pose their questions to our experts, both in German or English. And in our live stream, you will make a live chat tool available to you, so you can see it here. That's why we have the QR code here. So perhaps you could scan it with your smartphones right now, and then you can use that to enter your questions, and then they will be displayed here. So make research visible. So ideally, that is having an impact in society. That is the main aim of tonight's event. Professor Brigitte Röder, the organizer, a psychologist and neuroscientist at the University of Hamburg, has put together a very impressive program for us. And I personally am quite curious about this. My two sons are all grown up, and after everything that I will hear tonight, I will look back at the first years of learning of my children, and just like all the grandparents and parents listening tonight. So it's also about my personal peace of mind. Now we are looking forward to the presentation by Professor Dr. Hauke here, the President of University of Hamburg. And we are very happy to be here in this beautiful hall at the University of Hamburg. Good evening, or moin, as we like to say in Hamburg. Mr. Hector, members of the foundation, and dear Hectorians, as they like to be called, as I just learned. Who's part of that group? Okay, show of hands here. And dear colleagues, ladies and gentlemen, I'm really pleased to welcome you here as the president of the University of Hamburg. And I'd like to welcome you also in this stream. This is the 9th Hector Fellow Academy Symposium, and we are very pleased to be the host of this event here in Hamburg. Now, the aim of this annual symposium is to bring together excellent scientists together and to have an exchange with the general public to talk about the most recent findings and social and political issues. And as Ms. Grießmann just said, today we are talking about why do children and adults learn differently. And this seems to be a very fascinating topic. We will have many interesting insights, I'm sure, tonight. And this question, of course, relates to very important issues, everything relevant to the future of our society. And if I may put it this way, I'm a neuroscientist working in cognitive neuroscience, and in this role as president of the university, I see many opportunities here to build bridges in this symposium. First of all, building bridges between different disciplines. You will see this here, and also building bridges between research and society, and bridges between generations. And I believe it's not an exaggeration to say that the understanding of the learning of children, the factors that influence it, is the key to sustainable and just education for all. And we as a university, of course, are fully involved in this. And as I said, in my own research here, I worked in neural mechanisms of learning and decision making, also working with children. The most recent papers have just been published. How do children learn, for instance, by observation? And that's why this is a special event for me as well. And you can imagine we bring our experience into our roles, and it also plays a role in how I lead this university, that I've looked into learning and decision making, and that's why this is so interesting to me personally for all kinds of reasons. And we as University of Hamburg, we strive to design teaching and research here that it follows the natural learning process of our brains, and at the same time living up to the challenges of our times. And in the past 2 years, we as university, together with the University Medical Center in Eppendorf, which is working in research in neurosciences as well, we've expanded this research, and one institution represents this, especially the Hamburg Center for Neuroscience. And it networks here in Hamburg and the metropolitan region more than 400 scientists from 31 institutes and clinical departments, just to give you an impression of how many people are actually working on these subjects that we will discuss tonight right here in Hamburg. And this kind of interdisciplinary work and this kind of program that we will have tonight, this represents our vision of the University of Hamburg here for networking in research as a university. And today's subject is of very high social relevance, and from my point of view it's related to very critical aspects of what we like to call twin transformation here at the university. Here we were involved in two big transformations here. First of all, sustainability, and secondly, digitization. And we are convinced that we can only understand these if they look at the interrelation between them and we actually apply them in real life. And now the research results from these areas are very inspiring. These are innovative approaches in educational science, pedagogics, and digital education. And just to give you an example here, what I'm referring to here in relation to the University of Hamburg here, that we use AI tools like ChatGPT at the university intentionally. Of course, we take a critical perspective as well, but even months ago here, we rolled out something that we call University of Hamburg GPT, because we have developed methods for us that deal with this subject. And before I conclude, I'd like to announce a few words of thanks to Mr. Hector, first of all, for your wonderful commitment to science and many other things, but tonight mainly for your commitment to science and education, and the representatives of the Hector Foundation and the Hector Fellow Academy. It is really important that we have not just public funding to promote excellence in science, but that we have individuals like you who support education with their foundations, because only in this way we can defend our free democracies, because we offer education for all. So progress, innovation, that is something that we as a country need desperately, so that the good things that we have, that we promote them. And then I'd like to thank my colleague Brigitte Röder and all the other organizers who worked on preparing tonight's event. And with your work here, you show how interdisciplinary research can lead to a deeper understanding of human development. So, in summary, I'm convinced that the presentations and discussions that we will enjoy tonight will be very interesting and educating. They will send out impulses for sustainable and just educational environments. And that brings us to our common aim. With the results of research is to be translated into real life, so that we safeguard our future and design it together. And I wish you an inspiring evening. Enjoy, and it's great to have you here tonight. Enjoy this time at our university. Thank you for listening.
Professor Hauke Heekeren.
Now, Professor Hauke Heekeren, thank you very much for these words of welcome. And why do children and adults learn differently? I have a nice quote that Sebastian Frank has made available to you, our first speaker tonight, and you will meet him in a moment. He says, everyone can become the sculptor of their own brain. So what does a sculptor do? Well, they cut into the stone, they have different tools at hand, and this quote creates many images in our minds. So you can become the sculptors of our own brains. Santiago Felipe Ramon y Cajal, a Spanish doctor, said this. In 1906, he received the Nobel Prize for Medicine, and Sebastian Frank is a Hector Fellow Academy member and received the Hector Fellow Award last year. And he looks at neuronal mechanisms of learning in children. He knows whether it makes sense that children at an early age in kindergarten will actually learn Chinese. He asks, do children actually learn better than adults? And if so, why? We're looking forward to Sebastian Frank from the Institute of Psychology at the University of Regensburg. You have the floor.
Right, thank you very much for this kind introduction. Thank you very much. It's a big honor for me to be standing here in front of you tonight and to be able to talk about my research. So we're going to be talking about learning, and I'd like to get started with a brief reflection on just how important learning really is across the entire lifespan of human beings, both in childhood, during adolescence, during our professional career, and also later on when we're older. So because we preserve the ability to learn new things over our entire lifespan, so we never stop learning, in fact. And the question that we are dealing with in our group is whether mechanisms of learning change over our lifespan or whether they remain the same. In order to answer that, it's a good idea to first take a look at the basis of learning, which is the brain. So if you take a look at the brain over human lifespan, you will see that it is subject to a lot of change. Now, this is an example of how it changes on the y-axis. You can see the brain weight, and you can see that it rises dramatically, during childhood to the adolescent stage, remaining rather constant in order to go back towards the later stages in life. And the biggest changes occur between childhood and adolescence and adulthood. Right, the underlying assumption that many people have is that as the brain changes, our learning patterns change as well along this curve. During childhood it's pretty easy, they say, to learn new things, and the older you get, the more difficult will it become. The fewer things can you learn the older you get. And the question is, if that assumption were correct, shouldn't it be such that children can learn more than adults? And this is one of the questions that I'd like to discuss tonight together with you. Is it really so that children can actually learn more than adults? And the second question, which is directly linked to the first question, is what are the neuronal mechanisms that play a role in children's learning? In my research group, we're dealing with this question based on visual learning as a model. So visual learning, let me briefly talk about that. This is where the back part of our brain is responsible for the occipital lobe, which is indicated by this circle. I think you'll be familiar with visual learning from your day-to-day experience. Just think about radiologists who have gotten decades of experience looking at brain images, and whenever they see a new image that they've never seen before, a completely new patient, they can immediately tell, well, something's wrong here or something is quite right. So that's an example of visual expertise that has developed by learning. Or if you happen to be a stamp collector or coin collector and you've got lots of experience, I think you would probably agree if I say that, you know, I'm going to show you a new stamp, a new coin, and you'd instantly be able to say, well, is this a real stamp or is it a fake one? Or if you're interested in art, and if you happen to like one particular artist, particularly looking at this picture, which you may have never seen before, you will probably be able to say with a high likelihood, you know, who is the painter, who's the artist, simply by looking at the colors, by looking at the brush stroke, by looking at the subject. So can anybody tell, you know, who is the artist? Okay, great, spot on. So a lot of visual learners amongst you. That's great. Now, these were examples from our daily life. But how do we go about it in our laboratories? How can we study visual learning? Now, to answer that question, I'd like to introduce a little experiment. So please do take a look at this image, okay? And if I were to ask you, what do you see on this picture? You'd probably say, well, it's black and white patterns. Now, I'd like to give you a hint, because a figure is hidden in this image, and I'm going to show you this picture again, and probably you'll be experiencing something that we refer to as the eureka effect in science. You'll be instantly able to see the figure in this image. And if I were to show it to you, you know, 50 years down the line, you'll still see the face. So that's an example of quick and long-lasting visual learning. But it doesn't always go that fast, because here is another experiment. Now, what have we here? There's two stimuli on this image. On the left-hand side, left red, right green, and the other one is vice versa. The left one is the target stimulus, the other is the distractor stimulus. On the next image I'm going to show you a lot of distractors, but mind you, there's going to be one target, green on the right-hand side, red on the left-hand side. Please try and spot the target stimulus, and if you've found it, please clap your hands. So be prepared. You're looking for something that's green on the right-hand side and red on the left-hand side. Where is it? Okay. So the ones of you that haven't spotted the target, it's here. I haven't cheated on you, but you'd probably agree that this task was quite a difficult one. It's not as easy as the previous one with the face. In our experiments, we found that if you ask adults to participate in such an experiment, if they train that 1 hour every day, you know, the time to detect the target becomes shorter and shorter all the time. And this improvement in the search time is an example of visual learning. Now, I've spoken a lot about the underlying paradigms, the experiments that we are doing in my research group, but you may be wondering, what other methods are we using? Now, we do behavioral training, computer-based training. Here is one of our subjects doing a learning task, and we also use imaging technology, magnetic resonance tomography. We are trying to record brain imaging or brain activity whilst people learn a new task. Coming back to my original task, do children show more visual learning than adults? Well, we found that the answer is yes, they do, but they do so for irrelevant visual stimuli. Now, you may be wondering, what are irrelevant stimuli to begin with? You have to imagine this as follows. So you're sitting in a lecture hall wanting to listen to me. Now, it may well be, hopefully not right now, but theoretically speaking, you might be distracted by a lot of irrelevant stimuli. They make it difficult for you to not lose track of what I'm saying or what you can see on the charts. And this situation can also be simulated in a controlled environment. It works as follows. At the center of the screen, you're going to show irrelevant stimulus. And at the periphery, on the screen periphery, you're going to have all sorts of irrelevant stimuli. And we did so in an experiment with 20 children of elementary school age and 20 adults, young adults, control group. The task was as follows. So I'm going to talk you through the experiment. What you can see at the center of the screen, this is the relevant stimulus. Images of different fruit, vegetable, flowers, and animals. And the task of the subjects was to look at the center of the screen and to say, what animals are you seeing there? In order to make it a little more difficult, we decided to mask the stimuli. So they were masked, you know, by different colors. And you may wonder already, why is it that there's these small dots and arrows? These are the irrelevant stimuli. So coherent dot motion is what we're talking about, all moving in the same direction, presented at the same time as the relevant stimuli. But the task was not to focus on the irrelevant stimuli, only focus on the relevant stimuli, the animals, etc. In order to give you a better understanding how it worked, let me tell you the following. These are the 4 elements. A blue beetle, a red bird, a green butterfly, and a yellow fish. Please take a look at the center of the screen and try to find out what are the 2 animals I'm going to show you. Once you recognize an animal, if you think there was an animal, please clap your hands. There's going to be 2 animals that I'm going to show you, and please focus on the screen center and tell me who the animals are. Okay, it's difficult, I must admit, and I'm going to do it another go without the mask. Please take a look at the center of the screen and clap your hands once you've detected an animal. Sorry, I was going to show you something else. Great, great job. I'm going to run this sequence of images one more time. Please take a look at the visual periphery and try to detect in what direction the dots are moving. They will change the direction with every image. Moving to the right, to the top, to the bottom. Correctly spotted. Great, great job. So I think the underlying ideas become clear by now. Now, the design was as follows. There were 12 training sessions on separate days, each lasting 20 minutes, not that long at all. And before the training and after the training, we tried to find out to what extent the perception of coherent dot motion had changed. So how has perception changed for the irrelevant stimuli? And here's the results. 0 on the y-axis is the performance prior to training. Any values below 0 means that the perception of coherent dot motion is improved. Below 0 means that performance had deteriorated. So in other words, if the value is above 0, visual learning was given for the irrelevant stimulus. So these are the results for the adults. You can see there's no evidence for visual learning in adults. The opposite is true. The subjects had a deteriorating performance, probably because they were trying to inhibit irrelevant stimuli. Different story for the children. So the children are developing visual learning for the irrelevant stimulus. This was the result. Now, in order to summarize, it seems as if children finding it difficult to inhibit irrelevant stimuli, to suppress them, as it were. Seeing that they cannot inhibit them, there is more visual learning for the aforementioned irrelevant stimuli in children. Now, today we're talking about neuronal mechanisms. Now, you may wonder, why is it that children are finding it more difficult to inhibit these stimuli? Well, our suggestion is that this is to do with another structure, the prefrontal cortex, in the frontal area of the brain. And we know that the prefrontal cortex usually inhibits the processing of irrelevant visual stimuli in adults. Now, we also know that this brain structure takes a long time to mature until it's fully functional. It takes up until adulthood before this structure is fully developed. Now, as this structure matures, the prefrontal cortex, it becomes to link up. It links up increasingly with other regions. And at the very last stage, there's a link between the occipital lobe and the prefrontal cortex. Now, taking all these results together, you'd be able to come up with the following model. So, in adults, the prefrontal cortex is very well networked with the visual cortex, the visual parts, and inhibits irrelevant stimuli. So they're irrelevant, so it's a good idea for the brain to inhibit them. Whereas in children, that link is not fully established. The prefrontal cortex is anything but completely developed, which means that there is lower or no inhibition of the irrelevant stimuli at all. This is why visual learning takes place at a higher rate. The conclusion is that children do show more visual learning, however, for irrelevant stimuli. So that begs the question, and perhaps we can discuss that later on. Is it really so good to learn irrelevant visual stimuli? Probably not, I'd say. Just think of the fact that these stimuli are irrelevant, and it's perhaps a good idea to suppress them because they might distort and distract you from processing the task at hand. Is there anything positive to say about children's visual learning? Well, the answer is yes. I can tell you something positive. Children seem to stabilize visual learning faster than adults. Now, you may be wondering, what is stabilization to begin with? Now, let me tell you, whenever you learn something new, learning after you finished your training tends to be fragile to begin with, and it needs to go through various processes which we refer to as stabilization. So the idea is as follows. Let's imagine you train a task A for that matter. Once training's over, stabilization sets in during wakefulness and during sleep, designed to move your new learning results into a highly stable memory lane, as it were. So during wakefulness, if you were to train a second task B for that matter, what would happen? Well, training of task B can disrupt learning of task A, which means, and this is the consequence we've seen in adults, the consequence is that we do learn task B, but there's no learning for task A any longer. Now, what happens in children? We looked at that in another study, and I'm going to come back to it in a minute in more detail, but I'm going to preempt some of the results now. We found that children feature both learning for task B and for task A. Now, this is why we believe that the neuronal mechanisms of stabilization differ between children and adults. The question is, to what extent and in how far do they differ? Well, I'm going to try and answer looking at the following study. 9 children and young adults were taking part in the study. And before I continue, you need to know that learning, stabilization of new learning, involves the inhibitory neurotransmitter GABA, gamma-aminobutyric acid. And the concentration of GABA can be measured in human beings by way of MRS, magnetic resonance spectroscopy. And this is what the experimental setup looks like with one of our children. It works as follows. What you do is you take a photograph, an image of the brain, and you select a specific part of the brain indicated by the yellow rectangle. And within this rectangle, you measure a spectrum of different metabolic concentrations. Details are not that important. The only thing to remember is that there's one peak in this spectrum indicating the GABA concentration at the point when the measurement is performed. Now, with this methodology, we went for the following design. We measured the baseline concentration of GABA to begin with. Then we exposed the children to a visual training task, and once training was over, we measured the GABA concentration over again. And here is our results. On the x-axis, you can see the 3 measurement points in time, y-axis GABA concentration. Here's the adult result. We found that GABA concentration was relatively stable throughout the entire period. There was a bit of an increase towards the end, which suggests incipient stabilization, but it's only a very minor increase. What about children? Well, to begin with, pre-training, we found that they showed much less GABA, less inhibition in the visual cortex, which fits in with the idea that children may find it difficult to inhibit. But surprisingly, during training, and especially after training, we saw that GABA concentration showed a sudden increase to reach levels of inhibition in the visual cortex pretty similar to the ones in adults. So it seems as if the stabilization of new learning, the fast stabilization in children, is so fast because of the massive increase of GABA, which allows children to do more visual learning, which is why children show more visual learning and forget fewer things. I'm almost at the end, and I'm going to come back to the initial questions. Can children actually learn more than adults? The answer is yes, but this is not always positive because they also learn irrelevant stimuli. And what are the neuronal mechanisms that play a role in children's learning? Well, I hope to have shown you that it's inhibitory processing that's so important for learning during childhood. Right, that's it from me. I'd like to thank my group members in the top left side. I'd like to thank the Hector Fellow Academy. I'd like to thank Elsner and Hector, without who this great symposium would not have been possible. I'd also like to thank my cooperation partners in the US and at Regensburg University. And I'd also like to thank my mother, Brigitte Frank, who's also here today, without who I probably wouldn't have even started my research career, because, you know, she was the headmaster of a elementary school. We did a lot of these experiments, and without her, I wouldn't have been able to present these great data. Thank you very much for your attention. Dr. Sebastian Frank, thank you ever so much for this very instructive talk. I don't know what you feel like. It makes you feel humble, don't you think? I mean, you know, green, red, right, left, etc. Now we know why children are easily superior to us, you know, at board games. We're never going to win against them. Next presentation, ladies and gentlemen, is titled The Melody of the Immature Brain. Beautiful title. It's about brain activity. So much so good. Now, as you are about to listen to the next presentation, and let's just imagine you were equipped with the cables, you know, with an electroencephalograph. If we were to measure your activity, your brain activity, we'd see a lot going on probably, at least that's what I hope. And that would be pretty normal. This brain activity is primarily researched in adult brains, when it was really discovered in a child brain. Hans Berger, at the end of the 19th century, what he did was he, you know, connected his 10-year-old son to a number of cables to come up with a number of interesting results. We are looking forward to the next presentation by the head of the Institute for Development and Neurophysiology at the University Hospital Hamburg-Eppendorf, Professor Ileana Hanganu-Opatz. You have the floor.
Thank you so much, Ms. Wittmann, for this kind introduction. Mr. Hector, dear Hectorians, beautiful term, ladies and gentlemen. It is a great honor for me and a great pleasure as well here to introduce the melody of the developing brain to you. That is the rhythms that we find in the developing brain. And the fact that you decided to come here tonight and that you are listening to me right now and that you did not go to the swimming pool instead, and the fact that hopefully you will have fond memories for a long time of this event, all this is thanks to a very complex network of brain regions which are shown here in a simplified version. And you see these regions are everywhere, and you find them everywhere across your brain, but there are certain hubs here. The little box in the frontal cortex were already mentioned, and the region here communicate, and that makes possible here that we develop all these skills like listening, be attentive, remember things. And we are very good at this, you know, you see it here. You didn't go to the pool. Yeah? But if I didn't have you as the audience, but elementary school children or kindergarten kids, then we would see that we are not born with these skills. And as we mature, these skills mature as well. And the big question here is, now, how do these skills mature? What exactly is happening in our brains? So how is this network created that you see here during childhood development? And these questions haven't been fully addressed or answered yet. I believe you don't have to be a neuroscientist to see that there's a huge difference between this structure here and the human brain during the 23rd week of pregnancy. That's what you see here. And this structure, that is an adult brain. And it is generally accepted that the main structure of the brain here is influenced by genes and molecular factors, and that's created in this way. And in a later stage, influence from the environment play an important role. In Brigitte Röder's presentation, we'll talk more about this. Now, this kind of influence or impact create electrical activity in our brains. How does it happen? Well, the nerve cells, the neurons, can all be excited. So they are in two stages, either active or not active, meaning it's nothing other than a binary system, 0 and 1. But the question we derive from this is, how is this done here? Are they all synchronous or asynchronous? How do they interact, these neurons? And to show you this, I'm going to show you this film. So these are metronomes, as you can see, and these metronomes are moving back and forth in a random pattern. If you were to imagine each metronome is a nerve cell, and whenever they're active, you see this little uptick. And if you have electrodes in your head, as Ms. Grießmann said here, in the EEG, you could measure this kind of activity and you would hardly see anything. It would just be noise, as you can hear. Nothing, just noise. But, just by magic, it seems, here from starting on the left, these metronomes will start moving in the same direction, or the same rhythm. And if you take little ticks here, you're less random after some time, but following a certain pattern. What is happening here? You can hear it. A certain rhythm is being created. No longer just white noise, but there's a rhythm here. And that's the melody or brain rhythm that you see here. It's called oscillation, in its technical term. I will talk more about this. So this kind of oscillation, now you hear it clearly, yeah? So this kind of oscillation, as Ms. Grießmann mentioned earlier, were all found 100 years ago by Hans Berger and researched by him. On the 6th of July is when he did the research here, and he published on this, and most of the EEG measurements were done during adulthood. You will see this here, the rhythm, the oscillation, different frequencies, fast or slow. And if you listen closely, there are fast rhythms in your brain. If you're bored by what I'm talking about, then it will slow down the activity, and the sleepier you are, well, the slower the rhythm will be. This kind of activity was also measured in children. And interestingly enough, we did this very early, at a very early age here. That's a study I conducted together with colleagues in Marseille and Paris. What you see here is preemie in the 25th week of pregnancy here. That's good. This is the hand of the doctor teaching the child here in an incubator. And the activity is not continuous but discontinuous. You see a flat line for some time, and then you will see these little peaks here in the oscillation can be induced with the hand, tactile stimulation, or can also happen spontaneously. And it's not just tactile stimuli that you see here in this oscillation. We tried light and sound will result in the same kind of activity in the brain. Now these children are hardly bigger than my hand, and we can't do more than measure this activity. And the big question, however, is, so what's behind this? What kind of neural networks contribute to these activities? Why is it discontinuously or not continuously? And the biggest question is, does it have any meaning at all? Or it's just a byproduct of development? In order to address this question, you can imagine, I mean, you cannot really work with preemies. For ethical and technical reasons, you can't do much more than this kind of measurement, and that's why it's necessary to have an animal model. And we are lucky because most of the animal models, that's mice and rats, are born with a very immature brain. The maturation of the brain happens after birth. And so if you look at the maturation, development of brains here, and looking at two species, you see that the activities in this child here, that is in the second trimester of pregnancy, corresponds to the activity of this animal in the first week postnatally. And the rhythms activities are very similar, hard to distinguish. Very difficult to say which of these rhythms or these oscillations come from a human, or in this case, a rat. And that gives us the opportunity to address the question I mentioned earlier. What kind of mechanisms play into this, or what do you need in order to induce this activity, and also what role does this activity play? we investigated this and we found then, or confirmed, that the activity can be found in mice brains just like in a human. First it's continuous, and then the older the animal gets, more and more continuously. And depending on what the animal does, if it sleeps, it's just slow, or when the animal is working on a cognitive task and they're good at that, then you will see this kind of fast oscillation, just like when we pay attention. And surprisingly enough, here in this animal model, here we are measuring the activity not just like in the EEG. We can also see what does the individual nerve cell do in this overall network. And that's, you see on the lower part here, the individual lines. These are the responses. Here, the 1 in the binary system. So the 1 that we get from nerve cell when active, then you see this little tick here in the chart. And we use the information to understand how the networking takes place in the brain. And there's one big question we want to address with this. That is, what is the meaning of oscillatory activity for the development of the brain? Is it just a byproduct? In other words, the nerve cells will mature, becoming active, excitable, generating the 1, and then randomly we create this kind of rhythms, or are these rhythms necessary in order for the brain to develop? And we're mainly focusing on the cognitive skill of the brain with a special focus here, and I'm really thankful that Brigitte mentioned here of the prefrontal cortex. That is basically the control center of cognitive skills, not the only region in the brain influencing attention and decision making and others, but it's one of the most important ones. And what you see here in the prefrontal cortex here, we see in humans, but also in rodents. And they can also come to good decisions quickly. That's why it's so hard to fight. And we investigate the region of the brain, and in several studies we identified which nerve cells were responsible for different rhythms. What you see here in this diagram is so-called pyramid cells within a certain layer of the prefrontal cortex responsible for this cognitive activity. 16 to 40 hertz is what we see here. That's an activity. We know which cells are involved, and that means we can manipulate this activity. We can change this kind of rhythm to see whether it has an impact on later cognitive performance of these animals. And we did that. And we used a method that the symposium dealt with last year, the octogenetics, that is to manipulate, use light for the manipulation of nerve cells. So they're not just light-sensitive neurons, but you create light sensitivity by experimenting light-sensitive parts of these neurons. They respond to light, and you can activate them, excite them whenever you like. And that's what we did here. You see this procedure here, rodents, mice, 7 to 11 days old, and the proteins were expressed and manipulated them half an hour for 4 days, and we looked at the effect it had. This kind of manipulation resulted in these rhythms that and changed them that I showed you earlier. After stimulation, we found, that's not really surprising, when nerve cells are activated too much, getting too much input, how do they respond? They will then create more and more dendrites, more denser and denser network that's here stimulated and not stimulated. You see these little lines here, the dendrites and axons. And then this is the effect that will disappear after a short time, but then problems will start. 2 weeks after this kind of manipulation, the animals have already cognitive deficits. In these, what you can test with these young animals, so whether they recognize their mother. They were not able to do so. And what I'm showing you here is exactly this. Here, this skill to recognize their mother is part of the open fields in the short arena. Here, that had disappeared, and this ability had disappeared, and they had a weaker reaction to stimuli, and this effect was getting worse and worse the older the children or these animals got. So you look at the animals as adult ones, 40-50 days after birth, in other words, we will see that here the memory function is weaker, much weaker, and they make more mistakes here in memory tasks, and they do not recognize the other mice. So mice are very social animals. They always sit together. And after this manipulation, they will not recognize other mice in this little cage. Usually they would sit together, and here they no longer do so. And also, as I said here, the response to acute stimuli from the environment will be much weaker. And this means that here the change in activity is important. So you're not just changing the rhythm of the activity, it's also important when you manipulate it. And we're able to show here that so when you do stimulation 4 days later, it will have no effect. Meaning, yet our conclusion then that the oscillatory activity during development is critical here. It has to happen during a specific phase, otherwise you result in cognitive deficits and massive misconnection in the brain. It could be a critical period for cognitive development, and for humans then this early phase, it's around the time of birth, right before and after birth. But that's not the only critical phase. There's a second one, and that's later. It's a study we just published here. We were able to show the adolescent time window is the second critical phase. The children will say, I've always known about this. It's true. It's a time when the brain will respond strongly to stimuli, and here it's important to have it within the normal range. Why is it important? It's not just relevant to the normal condition of the brain, it's also important because these critical phases of brain development, these are phases of very high vulnerability at the same time. Our research has shown that during these phases, right before and after birth and during adolescence, negative environmental impact or influence can lead to massive changes in the neural networks and cognitive deficits or the symptoms here also of mental diseases. We're able to show that in the case of schizophrenia, where these two phases here around time of birth and adolescence were of major importance. And during our discussion, we can come back to that. You might say, okay, adolescents, you know, there's here the negative impact of bullying, for instance, or certain drugs. Here they will have a very serious impact, more serious than during other phases of human life. That brings me to the end of my presentation. I'd like to thank for the support in the project I mentioned here, like the Deutsche Forschungsgesellschaft, European Research Council as well. They were really brave and supported our research. We called the psychocell. They asked me, do we have psychocells? They asked me, and I said, I have no idea. That's why I need your money to do the research. And they gave us the money. And we found that these are the pyramid cells that I showed you earlier in this specific layer of the prefrontal cortex. I'd like to especially thank my team and cooperation partners for the years of research that resulted in the outcomes I presented today. And I'd like to thank you for listening. And I'm looking forward to our discussion. Thank you very much. Professor Ileana Hanganu, thank you so much for this fascinating presentation. That is the melody and rhythms of the immature brain. And now we will focus on early childhood experiences. How important are these? How important is what we experience in the first months and years of our lives? How important is this for our development? What kind of impact does it have? How does it impact the development of our brains? And how can you find out about this as a researcher? Professor Brigitte Röder is ready here, the scientific organizer of this event here, has concentrated in fascinating research approach. She's working with people who were born blind and regain eyesight by surgery later. And Brigitte Röder here from the Institute of Psychology at the University of Hamburg. You have the floor.
Good evening, ladies and gentlemen, Mr. Hector. Ladies and gentlemen, moin moin, as the university president already said. Now, let me start, ladies and gentlemen, by showing you a piece of art. Now, this is a piece of art that I've brought all the way with me from an Indian artist, Chippa Sudhakar. It's referred to as the Spectator. The Spectator, that's the name of the piece of art. Spectator is wearing a pair of glasses, symbolizing the environment, the earth, terracotta. It's a three-dimensional environment. So what does Chippa Sudhakar want to say, want to tell us? The idea is that perception is not a one-on-one reconstruction of the environment, but perception always involves a transformational process. In neuroscience, we know that this transformational process doesn't happen by way of goggles or glasses, but through our brain, and we've heard a lot of that already. So visual perception, as you all know, starts by what we see with our eyes. It all starts with a two-dimensional representation of the field of vision of our environment, and we all know that we've only got one image in our brain. So it's within our brain that the two-dimensional image is converted into a three-dimensional perception of the world around us. And during that process, a lot of things can happen. The brain can make mistakes. And that's why most of you will see a square, even though there's no square on the image on the right-hand side. So what's happening is that there's basically four angles that would form a square if, you know, there were a square, but we've learned that these angles always form a square shape, and this is prior knowledge that we use to be able to analyze our environment rather quickly. So we don't need to analyze everything from scratch, but we rely on prior knowledge. Now, in my presentation, I'm going to tackle the question of what is the role of early experience, prior knowledge, and what role does some early childhood experience play when it comes to the acquisition of neuronal structures that are responsible for these perceptual patterns. Now, how we do that? What's the experimental setup? Well, we've got to manipulate perception to begin with. Of course, we're not allowed to do that in children. We need to look for natural cases of manipulation. And this is why we've decided to research children or adults with congenital cataracts. You're all familiar with cataracts. It's a common disease, and you can see what it looks like in patients. So foggy or clouded lenses. If you suffer from that particular disease, it's like looking through a tinted or fogged up glass. So these children were all born blind, and at the Prasad Eye Institute in Hyderabad in India, surgery is performed on children with these diseases. And this allows us to study whether these children can still learn after having undergone surgery. The early developmental stages of the brain are already over at that stage when surgery occurs. And the question is whether the structural and functional brain development can be caught up on, as it were. In the previous speech we heard that there are sensitive periods, very tight, narrow time windows that are important, you know, for forming the adult brain. So what we've decided to do was to look at the structural development of the brain. Here is another one of these beautiful MRI images. It's a cross-section through the brain. And hang on, go back. Here you can see the outer shell of the brain. You know, there's some gray shape. It's the so-called gray matter of the brain. In the gray matter, you will find the nuclei of the neurons, which is basically the computer centers of our brain. Our brain is beautifully folded that way so that as many as neurons can be accommodated, which explains the great performance of our brain. Now, how can we measure this structure? Well, we're using MRI. Sebastian Frank, we talked about that, and by way of magnetic resonance imaging, you can also detect and record the brain structure, and it allows you to measure the thickness of the grey matter and also the surface. We've also seen images, brain images of embryos and also of adults. And what you can see here is the brain of a 1-week-old infant and a 2-year-old toddler. And as you can see, it's gained weight massively. If you take a closer look, you will see that the cortical thickness initially increases a lot. And that's also plotted on the chart in the bottom left corner. And this is due to the formation of connections, brains becoming wired. Then over time, these connections are pruned, the cortex becomes thinner again, and the assumption is that this process very much depends on experience. And I should also add that at the same time, the surface also grows. Now, how do we know that these two structural developments or changes depend on experience? Well, this was worked out by looking at patients or people with a congenital blindness. So they were born blind, their blindness could not be treated, and we found that the cortical surface is smaller, and at the same time thickness was pronounced more strongly. Sounds counterintuitive to begin with, but you can explain it by saying that the generation of wirings or connections is independent of experience. So it's a genetic program that is run, and then pruning happens as a result of environmental experience. So another critical question is whether this process is linked to early childhood development, or can you repeat it or simulate it at a later point in life? To answer that question, to try and answer that question, a study was conducted, and it was based on images of the adult brain or adolescent and child brain of people who suffered from congenital blindness, or of patients who were born with cataracts, who had undergone surgery to do away with the cataracts. And what Korte found in the context of her research was the following, and I'm going to go back one chart. You can see it here. As regards the blind people, the cortical thickness was higher, but that was also the case for patients born blind but who had their vision restored at a later point in their lives. Now, this suggests that there seems to be, sensitive periods for the structural formation of the human brain. And similar results were obtained for the cortical surface, as demonstrated here, expressed not that strongly, as you can see here. Now, the question is, what are the effects of such permanent structural changes on the functionality of the brain? Now, to answer that question, we did a functional MRI, and we started by looking at the ways of working, the organizational structure of the visual cortex. Now, this is the field of vision, and in the brain, it is represented by the primary visual cortex, and it's all appended. So the upper field of vision is represented the lower visual cortex, and vice versa. You will find the right field of vision in the left part of your brain, and then there's also a small share for highest visual acuity, which is represented down here. Now, this is retinotopic organization, and in the meantime, it's possible to make that visible by way of MRI images. This is an image produced by Karen Heitmann, who was responsible for conducting that study. You can see these retinotopy charts of the subjects. Now, after having obtained these images, you can drill down deep and look at the finer points of the structure. So what we did was we looked at the most minute structures to find out how much of the visual cortex represents how much of the environment. Similar to camera, you know, if you've got a high resolution camera, you will obtain much better resolution performance, of course. And if you've got a lot of, if your camera is even higher performing, the image quality will become even better. So the highest acuity central vision comes with a very high resolution, and the representation is in line with that. Now we try to find out what does that mean for patients who had their vision restored at a later point in time. Well, initially we found that there's also a type of retinotopic organization, which is good news. Seems like there is well-ordered processing in the visual cortex, also happening in these patients. But if you look at the reception, these pixels tend to be larger, and if you look at the representation, it will be lower. And that's not really surprising, because from earlier studies we know that the amount of representation, the size of representation, is directly linked to the structural surface of the cortex. Now, why do we need all that in the first place? Well, we need these structures in order to be able to detect faces. Whenever we look at faces, you know, we look at, well, the person in front of us, and we try to find out how does visual perception or facial perception work in subjects that were born blind, having had their vision restored by way of surgery. And the tasks they were exposed to were as follows. If patients were asked, who is the person, where is the person, you know, on the within the green square, it's not that difficult. All you need to do is analyze the size of the white block. And it's a little more difficult in the second task. Here, the persons are shown from the side, and in order to solve that problem, you have to turn the frontal vision to a side view of the person, which is a transformational step, and it's becoming even more difficult if you change the light conditions, as in the third example. And we know from our subjects that they find it rather easy to solve the first problem, but more difficult to solve the second and third one. Now, why is it that we, with normal vision, don't find that difficult at all? We can detect people from all angles, no problem. And to try and answer that, we've developed a, try to work out where facial recognition happens in the brain. We can do that by face processing, and we've heard already something about that. So the subjects were fitted with these EEG hats that measure minimal electrical currents during neuronal activity. So what we did from the brain activity, we tried to extract so-called event-related potentials. And we know that one potential, which we refer to as N170, is particularly strongly expressed if you look at faces compared to looking at houses or random images. And we also know that this particular potential represents or reflects the activity of the facial recognition areas. Now the question was, is it possible for us to detect the individual facial recognition steps in our patients? Well, it's beautifully shown here. The typical pattern for the control group, you know, high amplitude for facial recognition and lower amplitudes for detecting other types of stimuli. For the congenital cataract patients, different story. Now, to begin with, they also show N170, which is good news. They've got an active area in their brain to detect their faces. However, there is no differentiation between the different categories, which means that activity is even stronger for random patterns. What does that mean? Well, it means that early childhood experience is essential for developing what we call the expert system. You all know that if you want to become an expert in any given discipline, you have to give up on other things and focus on one thing only. And that's a principle that, the brain seems to be following as well. So people with the congenital cataracts do have a visual perception area that processes everything pretty well, but not as perfect as in healthy patients, healthy subjects. And as I come to the conclusion, I'd like to talk about, the importance of early childhood experience for neuronal development. We believe it's important for the structural makeup and the structural formation of the human brain, based on which the functional refinement of the visual cortical processing takes place, because this ultimately allows us to effortlessly detect and recognize a whole lot of different shapes and forms. I'd like to conclude by thanking my excellent working group that have gone to great lengths to come up with all the results I've been fortunate enough to present to you today. I'd like to thank our cooperation partners and at the LVP1 Institute in India, Vagar Khan, Ramesh Kegunaya. I'd also like to thank our cooperation partners at Maastricht University and Lucid Medical Diagnostics, where we did the MRI imaging experiments. Thank you very much for your attention. Professor Brigitte Röder, tonight's host of this great symposium, thank you very much for this most exciting presentation. Now there's going to be one more presentation which will look at a completely new angle, because how can it be, or is it possible at all, that what my grandmother once experienced would still have an impact on what I do and think today? No. May well be, because I spent a lot of time with her. I talked to her a lot. She told me a lot of what she experienced when she was a child. But may it also be that my grandfather, who may have suffered hunger, but who I never ever got to know, also impacts who I am today, because I just love eating. Whenever I go, wherever I go, I have a bite. Is that possible? Is that possible? May it be possible that this is due to my grandfather? Well, the answer is yes, indeed, says Professor Thomas Elbert, who for 30 years now has dealt with epigenetic characterization of trauma and violence experience. Very interesting topic, very much looking forward to your presentation. Thomas Elbert, Professor for Clinical Psychology and Neuropsychology at Konstanz University. You have the floor.
Mr. Hector, ladies and gentlemen, now why do children learn differently from adults here? What are the reasons? We've talked about the mechanisms a lot, but now what's happening during evolution in this whole construct? And what you see here is a caterpillar as a child going through the bush here, eating the leaves, and then looking for a partner. Then it would nicer to have wings, and then they become a butterfly and have wings. Now both animals have the identical genetic makeup, genetic information. It can be a caterpillar, at the same time it can have wings and be a butterfly. How is it possible? Because it selects which genes to express and during which phase during their lives. And all of us here, they've already gone through this, sometimes painfully, that as we age, our gene expression changes. Some of this is pre-programmed. Looking at the age line here, and looking at the gene expression or the readability of genes, then we see a clear correlation. So from a drop of saliva, we can generate 850,000 markers, molecular biological markers that we can read out from your saliva. It contains here your DNA, and you can say which genes to express and which not. the different mechanisms playing. The one I talk about, it's a methyl group that we added, and if it's there, it's not read. Out of the 850,000 piece of information, I can take 150 or 200, and from that I can construct here the DNA MH, and you see clear correlation. Each circle represents a person research at the age of 40, for example. There are 10, 15 years of difference in expression. For instance, some people will have gray hair at this age, and the main difference is the adaptation to a potentially dangerous environment, how much stress I go through, and this will change my genetic age. And stressors have a major impact during our childhood, our youth, that prepares us for life in a dangerous or not so dangerous environment. And we already know here, if you're subject to physical or sexual abuse, 50% of girls in Germany, 6% of boys affected here by sexual abuse, and or physical abuse is very prevalent. And into emotional abuse is just as toxic. So this is a factor, but broken homes, for instance, here, household dysfunction and other stresses that impact children, they will determine which genes will be read and expressed in a later stage. It's an adaptation to our environment. If you have 5 or 6 of these items can be checked off your personal list, then there is a certain probability, the 25%, to attempt suicide at some point in your life. If there's 0 that you check, then the probability of suicide is basically 0. And in addition to these household dysfunctions, then if you look at the greater world here, these are data from last month, June 24. That is wars and other events. So which countries are involved in wars? And if you are in Kiev or Tel Aviv, you are under constant pressure, under constant threat. And children are trying to adapt to this kind of dangerous environment. And we can also show you what the picture looked like in Europe. Where was there violence here? Conflicts, violent conflicts in Europe last month here, and quite a few, if you see this map. So we are supposed to adapt to this kind of environment depending on how dangerous that is. And our main defense mechanism, sort of the elite force of defense, is this HPA axis. That leads here from the hypothalamus in our brain to the pituitary gland, and from there moving on here, the chemical signal, you might have heard about it here, to the adrenal gland here. That's the cortex of the adrenal gland, and it will then emit cortisol, and that's the fuel, the energy that we have here. We activate it in the mornings here by emitting cortisol, brings us through the day. Then in the afternoon might be a slump, but whenever there is danger looming, or when you listen to this wonderful presentation, these tonight here, we will also emit more cortisol, of course. And if it doesn't work by my presentation, I would have to pose a threat to you, which would be extreme. So when the risk of danger is over, then you have receptors which will inhibit this activation. That's a healthy situation. And this is, of course, a complex process, but we don't need to look at the details. And But what I wanted to say is that these receptors can be controlled, and the gene expression of these receptors can be controlled, regulated. So if this HPA stress axis works well and correctly, then we can deal with threats. But if there is continuous threat, like bombs or the drunk father coming home, will he hit me, will he kiss me? If you are under these kinds of threats, then you will see that this axis is being activated time and again, going up and down, and this will result in a situation where our systems are no longer really regulated. The immune system is then restructured, remodeled, basically, adapting to the risk of injury and not responding well to a typical infection, meaning that yes, this was just to activate the cortisol in you. That's why we dropped this thing here. Some people who will be scared easily, then they have a different HPA reaction here based on previous experience. So depression or infection, it's bad, both of this. And when we end up in fatigue, then we end up with something we don't want is here, that we lose muscle matter and abdominal fat is increased, not what we want to see. This HPA information, the cortisol and the activation of the HPA is also something that a mother experiences. 20% of women will experience violence by their partner, and if it happens during pregnancy, this will mean that by way of a very fine, well-regulated cascade of biochemical processes, this child here in the uterus was also being informed here, like, be careful, you are at risk, or risky environment. And this information, this readability of genes is already modulated in a child at this developmental stage. And Mr. Hector, we work with Axel Bayer, another Hector Fellow, and his team, and we looked at this, what happens when a woman during pregnancy is subject to intimate partner violence. And what we see is, this is here from one of these clones here, then the methylation, the readability of genes is changed. And now we have a lot of research. Fernanda Sapoloni mainly worked on this. Also, we know there are many genes that can be impacted. The child will develop differently in these situations. And we've also seen that when many adverse experiences are impacting a child and the readability of genes is changed, then we can end up here in personality disorders expressed in different ways. So depending on how you're prepared as a fetus here, what kind of bad experiences, adverse experiences you had before birth will impact development of your personality. So like narcissism, but Machiavellism, things like that we also see in the Russian president. This is how it can start. And the question is then, how can we cure that or modulate that? Here, together with Fernanda, we worked in the favelas around Rio de Janeiro, that is the poor regions. And there the grandmother is 60 years old, mother 35 years old, and the child 10 or 15 years old. So you look at this across 3 generations, and you see very high variance across all the threats and the dangers that these people have experienced. So there are women who see how others are killed, who are threatened or beaten, and others who live in poverty but still have a fairly good life. So there's a certain variance. And the result, don't be scared, but looking at this, it looks very complicated. We talk about 800 different loci and all the correlations, but we've broken it down for you into these simple regions. The first point is not surprising. Mothers exposed to intimate partner violence during pregnancy, well, be it at home or in organized fashion, have a increased risk of depression. And if they have enough of these adverse experiences, they will develop PTSD and show anxiety symptoms. And children, when they have the same experience at a later point in time, will see the same results. But what was quite surprising is, now, if the mother experiences this kind of danger during pregnancy, she programs the child, probably via the HPA axis. And the child is reprogrammed in such a way so that it will not suffer so much at a later point in time from these mental diseases. And we see this here also. The FKBP, so the modulation in the HPA axis. And we can also see it here in the water flea here. We also see it among fish, and we see it here in these fleas. So the mother, when it grows up in an area with a high density of predatory fish, then the offspring will show spikes here and helmets, and then they're not as tasty to the predatory fish anymore. And they also change their behavior, and all of a sudden they're not willing to swim to the surface during the day, which would be a good thing to do. If there were no predatory fish, they would do so. And humans who have this experience of growing up in a dangerous environment, or might already have received danger signals in the uterus, will not be born with spikes on their head, but with a different neural regulation, different stress axis, and will be fast reactive and stronger appetite aggression, but that might be better in a favela environment, can respond to threats faster and stronger. And we also find when these people have used violence once, they will get more positive feelings and positive experiences from using violence. And we know that drug consumption, drug abuse is very prevalent. If someone were to say that it's just a correlation that you are reason you are not manipulating the environment systematically, can tell you that's, of course, you can only do with animals. And here, Stephen Somi on the other side of Lake of Konstanz here, where you have these animals, these monkeys, and the mother will carry the young one on her back. But these monkeys, can they grow up without a mother, just with their peers? So what you will find is that they have a different type of cortisol regulation, different regulation of the complex HPA system. So they will have bonding difficulties, just like if you worked with orphans. The children will just come up to your lap. I mean, that's a disruption in their bonding abilities. They should be cautious. And children are hyper attached, more fearful, more aggressive, and they also show a predilection to alcohol abuse. And they also have a changed immune system. we see more cancer and more infection among them. Now, without going into detail, I wanted to show you one animal is shown here with different loci here in this status. And you see the animals on the right-hand side, those which grew up with their peers only show a different pattern than those who were raised by their mother. And how many of these genes here, made up of 30,000, 25,000 different proteins, 20,000, says Mr. Axelmeier, whatever. Okay, there are 20,000, some 25,000, some others, but differently regulated in this kind of environment. Now, how does it change? Like among these little monkeys, 5 to 6,000. And what is regularly different? Our immune system and also our neural network. And what does it mean for us that if I've had many adverse experiences during childhood or adolescence, I'm better suited to survive in a dangerous environment, but in a peaceful environment it will cost me 20, 25 years of my life because I'm more susceptible to illness. And the last point I want to say, as Ms. Grießmann mentioned here, so if you look at this, you might say here a daughter is growing here in this uterus, and already in uterus here in the germ line here, this is is already created. The egg that you came from was created by your grandmother, basically, or influenced by your grandmother. According to our calculations, it's 10 to 20%. This influence, much less than the situations experienced by parents, but still there. And also in animal experiments you can show this. And statistically speaking, it's not that easy because there are so many dots here. You can't just say the probability is 1% or 5%. You know, you have to look at 10 to the power of -6 or -7 probability, then I'm statistically sound. And if I take the highest elements, then I end up with these changes in the circulatory system, what you see in PTSD and depression linked to all of these effects. And now I hope that you've received all the right information when you were still in utero, and get all the right information from your grandparents, and will live to a long and healthy old age. Everything else might protect you from dangerous environments. Adaptation to dangerous environments, however, is not healthy if you live in a peaceful environment. So why do children learn differently, different from adults? They learn to adapt to their environment. And after this adaptation has happened, so when you've grown wings as a butterfly, you cannot reverse the process. Thank you very much for listening.
Thank you very much. Why don't you stay on the stage? Thank you very much for this exciting presentation. Now, I've been reflecting my family history, you know, to the extent I know it. Perhaps some of you may have felt similarly. Thank you very much for the questions that we've received by way of the live stream, by the way. I'd like to ask all the speakers to come to the stage. You, distinguished members of the audience, may ask questions using the question cube that I'm happy to throw to you. Now, before we get started, I need to inform you that Professor Martin Korte is otherwise engaged. He cannot be here with us tonight, unfortunately. That's why I'd like to start with you, Mr. Frank. It's one of the top questions. You said that there are so-called irrelevant stimuli that children are exposed to. So who decides what constitutes irrelevant stimuli, what are relevant stimuli? Well, that's a decision to be made by the teachers. That's defined by teachers. They define what's relevant, what's not. Sometimes we do it ourselves by focusing on particular things that are meaningful to us, that distract us, that pop up unexpectedly, or because they may be so salient, different from the environment. So it's sort of an interplay of what we tell children, what's relevant, what's not, and what children are actively looking out for. And a follow-up question to that: from what age onwards can children hide or mask these irrelevant stimuli, or turn a blind eye to? Now, how do you define turning a blind eye to? Well, if you're defining it as suppressing because it's irrelevant, well, I'd like to say that based on our past experience, this becomes possible with effect from the age of 18 or so. We don't know what it's like in adolescence. Interesting question. Perhaps, how do adolescents, teenagers deal with the suppression of irrelevant stimuli? But I think it's fair to say that, from the age of 18, 20 onwards, are we in a good position to suppress irrelevant stimuli. Bad news is, however, that as we age, it becomes more difficult to suppress these kind of stimuli. Well, that's the sad news that I can't spare you. So starting with the 60 to 65, it's becoming increasingly difficult to suppress irrelevant stimuli. Interestingly enough, people of that age are featuring similar visual learning patterns as children. Questions from the audience? There's a question, and please do stand up and who you're putting this question to. I'd like to start with the same speaker. Here's a question for you, Mr. Frank. It's to do with the GABA mechanism that you elaborated on. And what about its role in different learning success, learning outcomes in children versus adults? You showed us that the GABA concentration is initially lower with children and rises during the problem-solving activity. This, however, doesn't tell me why GABA should play a role in more effective learning, because it's higher, as I understand. The concentration is higher, I understand, in adults. So perhaps you can explain, how to make sense of that. Good question. In adults, GABA does rise the longer you measure. In our experiment, we only measured or looked at the first 20 minutes after training. However, if you were to continue measuring hours after the end of training, what you'd find in adults would also be an increase of GABA. Perhaps not that pronounced as in children, but also quite significant. And I think this would also impact or reflect a successful stabilization of learning in adults. It's not like adults cannot increase their GABA concentration. I think this was what your question was about. Rather, GABA, well, you have to conduct longer measurements. You need to know that in adulthood, the GABA rise happens more slowly than in children. And looking at the end of life, currently we are performing measurements on older people with a similar task, trying to find out how does GABA concentration change at the end of training. And interestingly, we've seen that there's an excess of GABA in older people after training, much higher than in adolescents or children. And during training, or because of training, GABA is reduced, going down towards the end, only to rise again. Probably that's also reflecting learning stabilization activity. So GABA does change, or can change, over one's lifespan. The increase per se happens most strongly in children, and that this may be to do with the superior potential of showing such patterns, but we also see it in younger adults and adolescents. Are there more questions from the audience? There's another question here. My question is for Mr. Elbert. You spoke about different trauma children can be exposed to, sexual abuse, for instance. And I'd like to know whether this can be healed also in adulthood, whether such trauma can be healed during adulthood. Now, that's a very good question which hasn't fully been investigated, let alone fully been answered. We know that trauma symptoms can be changed. We can reduce trauma symptoms. And we also know that there are molecular biological markers that can normalize, but then there's also markers such as you can measure DNA repair. It can improve you know, it can go back to stages similar to non-traumatized patterns. But then there's also patterns where people align their performance, but there's also other patterns which causes people to become more different. So when it comes to gene expression, it's not like you can simply, you know, undoing a trauma. And yeah, from a neuronal perspective, this cannot be measured. You know, you can no longer forget it anymore. Now, telling someone that has experienced trauma, just forget about it, that's not possible. It will only aggravate the situation. It would only highlight it, make it come back stronger. Now, how trauma can be healed, what's good, what's not, very difficult to say. But then you also need to know, you know, if you think that what's most important for a human being's biological survival, for their biological fitness, is dealing with threats. I mean, where would you like to go? What would be the ideal environment? Are we going too far in Germany, you know, engaging in this threat hysteria? Is everything so dangerous as we pretend to be, or should we rather try and spike up our HPA axis? This is what we find difficult in trauma treatment. Are there further questions from the audience? If that's not the case, yeah, there's a question. You have the floor. Now, my question is to do with the hippocampus, which featured in Dr. Frank's presentation at the beginning, and it came up again during Professor Elbert's presentation. However, the hippocampus's particular role was not further expanded on. Now, I know, many of you may know, that the hippocampus plays a role in misfolded proteins, the fibrilla, that the hippocampus suffers from these kind of phenomena. And I'm wondering whether it's possible to find out in children whether the hippocampus is structured differently compared to adults. Want me to take that? Asks Dr. Elbert. Well, the hippocampus goes through a rather characteristic development. At the beginning, it is not fully developed. We cannot, episodically speaking, we cannot remember when and what happened to us, you know, before the age of 2. If someone says, well, I remember well how at the age of 1 I fell out of the pram, I must say that this kind of autobiographical memory was not formed at that time, because the hippocampus wouldn't have been mature enough. The hippocampus is something like the conductor of our memory, if I may put it that way, telling you where something happened, when something happened. And this capacity develops, and please, Frank, correct me if I'm wrong, the hippocampus, you know, takes until the age of 9 or 10 to become fully developed. Before that, you've got infantile amnesia. So the memory function, or the function of forgetting, is different in children compared to adults. And that might be important for your question. The hippocampus can create and form new neurons. If you regularly engage in sports, if you go for a walk regularly, the hippocampus, even at your age, will generate 5 to 10% new neurons. However, they don't survive unless you train them. They're going to be gone 1 year from now. So sports and the Hector Symposium, listening to a presentation like this, is going to be good for you. I'd also like to come in here, if I may. The interesting thing to know is that the hippocampus, we talked a lot about the prefrontal cortex, but truth is that the prefrontal cortex matures pretty late. We've heard that before. And maturation is driven by the hippocampus, you know. There is this excitation in the brain, and the prefrontal cortex acts as the master, triggering the input. Well, this goes back to the hippocampus activity. So the hippocampus is sort of the master commander, lurking in the background, making sure that the prefrontal development takes place with all the consequences we've seen. Very important. There is hippocampus activity, different types of activity, which is extremely important for the prefrontal activity. As regards some early childhood amnesia, it's interesting to know that there's a number of studies which suggest that children don't really forget. It's just not activated. You know, these memory lanes must be somewhere hidden deep down in people's brains so as to never surface again. And there's a string of studies out there that suggest that it might be possible to activate them, but this is still very much in its infancy, shall I say. We're going to do the symposium over again a couple of years from now. Remains interesting. Now, there's a question that we received from the live stream to Professor Röder. What about facial blindness? Is this a congenital defect, or is it to do with early childhood development? Well, there's two different types of facial blindness. There's developmental facial blindness, which exists from birth, basically. And we still don't know what the underlying root cause is. Why is it that people can't recognize faces? Compared to acquired face blindness, we are not aware of any morphological changes, you know. Probably, that's the hypothesis, is that congenital face blindness is due to successive miswiring in the brain, perhaps. So it's a functional but not a structural deficit, as in acquired face blindness. Thank you very much. We got time for one more question. There's another request for the floor. Please wait for the microphone. I was going to throw you the cube. Would have loved to do so. Anyway, I got a question for the two ladies, having engaged with face recognition. Now, in Japan and Singapore, they're using a lot of robots to do face recognition. Can I ask you, are you aware would it be possible, you know, to use similar approaches for people suffering from dementia or Alzheimer's, you know, given the shortage of skilled workers? What about robot-based face recognition? Do you think that's a promising avenue? So is it a possibility, because we spoke about ChatGPT earlier, you know, I'm asking to what extent AI might play a role. Now, I'm not talking about ethical aspects, of course. Now, I might as well give it a try. Says Ms. Hanganu-Opatz. These neuronal networks, of course, they are trained based on human face recognition patterns, and these neuronal networks basically reflect. I mean, we all know, you know, these queries on our computer screens. Are you a robot? Yes or no? This trains neural networks. Now, whether or not such patterns can be used for treating humans or as artificial limbs, as it were, you know, for people who have problems recognizing faces, as far as I know, there's no studies. Also, it's very difficult, very complicated. So I'd rather argue in favor of coming up with visual perceptual learning structures, what we just heard about, in order to train our natural networks. Because, you know, even if you know that they have been changed for good, you can still add or attach backup functions, as it were. And research is currently focusing on these kind of avenues, you know, using training patterns to modify altered networks, even though you need to know that it's never going to be as functional as a network that is fully intact and was trained, you know, from birth. Can I also come in here? These artificial neural networks, they're all based on excitement, but what they are lacking is inhibition. Whereas the brain, everything our brain does is coordinating interneurons, inhibiting neurons. And this is how far artificial and natural networks differ. Now, there's a new research avenue which says that artificial networks still have a lot to learn from the natural ones to master the skills and capabilities that we have. And when we're going to get there, no one knows, perhaps in a couple of years from now. Thank you very much. Unfortunately, we've reached the end of this talk. Thank you very much for the questions from the audience and also the questions we've received via the live stream. I'd like to thank you for your attention, and I'd like to hand over to Dr. Judith Elsner, the Managing Director of Hector Fellow Academy.
Thank you very much. Ladies and gentlemen, the Hector Fellow Academy has the aim to look at scientific issues and subjects and to present these in a way that is accessible to the broader public. And I believe we were able to observe that today. And I'd like to thank all our speakers for their input. It was very much an enlightening symposium dealing with a very complex subject. And I believe I can say we all will take something home from this and from these presentations. Ms. Grießmann was our host, conducting us through this wonderful event. Thank you very much. Without your assistance, it would have been difficult, I believe. And of course, I'd like to thank Hans-Werner Factor and the foundation, because without their support, we wouldn't be able to organize these symposiums. A big thank you for your long-term support to talk about all these subjects and make it understandable. This is something that will lead to progress for society. And I'd like to thank our host, the University of Hamburg, for having us here tonight. Thank you very much. Now, before we say goodbye to the live stream, I'd like to invite the audience here in the hall to a reception down in the lobby. You can continue discussion there. And let me mention that we have another subject that we'll deal with next year, where we will be in Düsseldorf. Professor Amunds, a Hector Fellow. Katrin Amunds has invited us to Düsseldorf. It's something that is related to tonight's subject. We'll talk about the brain, the computer, and AI. Will they soon be indistinguishable? And that brings us back to the last question we received from the audience. It seems there are mechanisms out there that give rise for hope here. Of course, we need to follow up on that, and we're looking forward to the symposium in Düsseldorf to learn more about this. And you're all invited to join us there on the 10th of July, 2025. Come to Düsseldorf or join us in the live stream. And at this point, we've reached the end of the symposium. I wish you inspiring discussions at our reception. Thank you very much for joining us tonight, and thank you for this lively discussion, and all the best to you. Thank you very much.